Welcome to Brain Freeze, the Dartmouth Undergraduate Journal of Science spring issue release event. Thank you all for coming. So I'm Hannah Payne. I'm the president of the DUJS, and for those of you who don't know what the DUJS is, it stands for the Dartmouth Undergraduate Journal of Science, and we're an entirely student-run undergraduate science journal. We basically put out one print issue every term. The issue this term is called "This Is Your Brain," and if you haven't seen it, it looks awesome. So thank you to all the DUJS staff. It's all cool. And if you haven't seen it yet, please look at a copy outside in the Great Hall. We also have a website, DUJS.dartmouth.edu, where we cover science news at Dartmouth, including events and publications. And part of our mission statement, officially, is also to facilitate interdisciplinary scientific discussion around campus. So that's basically what this is, all different types of science. And without much further delay, I would like to turn over to Professor Paul Whalen from the Psychological and Brain Sciences Department, who's going to give a really interesting lecture about the amygdala. And after that, we have a liquid nitrogen, we'll get some volunteers, so get ready to help out, and we'll make some ice cream. Thank you.
Thank you. I don't think I need that. I don't know about interesting, but well, good. You all seem to know each other already. I don't know how that happened, but I'm Paul Whalen, and I do brain imaging research in Moore Hall. We're a psychology department that's lucky enough to have our own brain scanner. A bunch of you have probably been in it. Well, since we're talking about journals and journal editing and research, I thought I'd start with a little journal story of my own. So what you do in this field, once you have a study that you think is interesting, you send it out to a journal. It could be an undergraduate journal on your own campus, or it could be a national journal. This journal is called Cerebral Cortex. And what happens is, if you get your paper, it's reviewed by your peers anonymously, and they decide if the science is up to snuff. And if it is, you get accepted, and then you get proofs, where they type out the whole thing for you, and you make sure that it's all accurate. And so when you are dealing with this one day yourselves, you always want to spell out an abbreviation before you use it. I know what Pavlovian conditioning is. I know a conditioned stimulus is a CS. And I know an unconditioned stimulus, previously neutral, is acquiring the affective value of a US. If you don't spell it out, they're going to spell it out for you, and it may end well or not end well. So the point here for the Hannahs of the world and whoever else is working on this journal, when you're editing, you got to know a little bit about your subject matter, or you're going to get a couple of things wrong. I think whatever you guys decide to study, for me it's psychology, you should do it in a place where something really cool and related to what you want to study happens. So the geo here is geography. I study affective neuroscience, or emotional processing, and I do it in Hanover, New Hampshire, because I don't have to go far to find a 24-year-old gentleman in 1847 who looked something like this, and only a year later, 39.1 miles away in Cavendish, Vermont, he would look like this, and end up being the most famous case of brain damage in the field of emotional science or affective science. This is, of course, Phineas Gage. If you go skiing at Okemo, you have to drive through Cavendish. And if you do, you will find a marker telling you all about this event. And as you may or may not know, he was incredibly appropriate and incredibly responsible. And after this 3.5-foot tamping iron entered his skull just behind his left eye and exited out the top of his head, he lived and became incredibly irresponsible and inappropriate. And so we're going to talk about some unrelated brain studies that go on in my lab, tell you a very short story about them, and then think about what happened to Phineas and why things like this occurred from his damage. I started my career working with rabbits. I somewhat naively maybe thought if I could learn and understand a rabbit brain, I might have a shot at understanding a human brain. I did that for 6 years through my PhD, and then for what's called a postdoc, after, what you can do with a postdoc is you can respecialize. you can go get another skill set. And so that's the story I tell people. What the truth is that I'm terrified of rabbits, and I didn't want to work with them anymore. It's just a personal choice. So I decided to work with humans. And instead of playing tones that predicted aversive outcomes, which is what we did with the rabbits, we decided that there are similar signals in the human world that predict outcomes for you. So if a tone that predicts a shock is a conditioned stimulus, and we want to call it a CS, so is a fearful expression on a person in front of you. They've detected something in the environment. You would do well to learn what that is. An area of the brain that turns out to be incredibly responsive to faces of others when they are afraid is the amygdala. It's buried. You have two of them. They're buried in your middle of your temporal lobe. So this is obviously a brain facing this direction. If I then slice through and turn the brain towards you, you can see the two of them there. The word is a Greek word that means almond. That's because of the shape of the structure in the coronal plane. Silly slide, I know, but if you take them away, so now you get a good sense of it's the gray matter there, buried in the middle of the temporal lobe on each side. I can give you a nice 3D cake slice image so you can see in context. Your amygdala probably reaches about this far back, and it's probably right here. What we do is we put you in the brain scanner here. We find out that your amygdala is incredibly active when you see faces like this. Your amygdala is doing this without your permission. It's not the fact that you're sitting there saying to yourself, that face looks afraid. We know this because we can present fear faces to you for only 17 milliseconds and then follow them with a neutral face. If we do that quick enough, what you'll tell us you saw is the brunette, and you won't report seeing the afraid face. But your amygdala will. We don't even need to show you a whole fear face. Amygdala is not that bright. It's probably not computing that whole face. We can just take out all the information but the size of the eye whites and run the study again, and you get the same exact effect. Amygdala is wildly active to fearful faces, masked fearful faces that are subliminal, that you don't know are there, and masked fearful eye whites. So it's pretty good at what it does, and its job apparently is to automatically do this for you all the time, to pick up on these signals even before you know they're there, maybe to get the ball rolling, maybe grease a few reflexes so you're ready for whatever it is you're going to do. The things that the amygdala can be sensitive to are incredibly subtle. We showed people pictures that looked like this and then showed other people pictures that looked like this. I don't know if the lighting in here is good enough for you to see that her pupils are a lot bigger here than they are here, and we've manipulated that. No one subject gets to see both pictures. They get one version only, and there's a bunch going by. Nobody reports noticing that pupil size is changing across the pictures, but amygdala does. It tracks larger pupils. What everyone thought the study was about, they said, I don't know, something about the hair, and that makes sense because we got these pictures from one of those hair cutting books, as in you too could have this hairdo. So that was kind of probably a nice distractor. We also used only male subjects and only female pictures because pupil dilation has a checkered history with attractiveness ratings. That is, some studies, about half only, will find that people report that pictures with larger pupils are more attractive. We measured that in this study, and we didn't find that. So we don't think that's what amygdala is tracking. And you know, that whole history comes from the fact that a long time ago, women used to put belladonna in their eyes and dilate their pupils on purpose. You know that, right? They thought it made them more attractive, right? You're going to the ball. There's going to be suitors there. You throw this stuff in your eyes. You're prettier. You get married, right? The problem is, if you've ever been to the eye doctor and gotten the eye drops to dilate your pupils, you realize you also can't see anything. So what ends up happening is a lot of pretty girls end up marrying a lot of really ugly guys, if you think about it. The point is that that's an incredibly subtle signal. You don't have to be aware of it, but amygdala is aware of it. When you dilate your pupils, it means you're interested in something. You've just detected something. If you've done that, and I'm close enough to see your pupil size, I might want to figure out what that is. The second point is that this all goes on without your permission. You can prove that these systems are separate in the brain. Emotional systems that function without your awareness, and other systems that are all about what you want to do voluntarily. We call that a cortex. So if I tell this gentleman to smile, he obviously has a lesion somewhere in his right motor cortex because he can't move the left side of his face. He's trying to, but he can't move any of the muscles over there, right? But then all I have to do is crack a hilarious joke, and he's fine, right? So that tells you that The automatic emotional systems that have access to the facial muscles are separate than the voluntary, explicit is the word we would use, motor systems that move the face when you want to move your face. Your facial movements are incredibly automatic. If somebody smiles and we're filming you, we'll show you that you start to mimic them, right? Very automatically. And I know, and you differ the degree to which you do this. So I know there's at least one of you sitting there going, yeah, no, I see people, I don't do that. Everyone does this. If I put electrodes on your muscles, I'll show you that you have a potential change in the zygomatic if I smile, right? If two people start kissing while you're watching a movie, you will start going like this, right? And everybody turns their head to the right. I don't know why that is. So tracking emotional expressions on the faces of others is important because it predicts outcomes for you. But the faces themselves predict things too, right? You have to learn about each other to navigate a social world. And so does the system that responds to tones that predict shock and facial expressions of other learn that some people are not very nice, and other people are very nice, and yet other people are rather boring. And when you're walking around the world, you want to know who to approach and who to avoid. So you have to learn about these things. And the question is amygdala part of a system that does that? I hope you're seeing what we're doing here. This is simple Pavlovian conditioning. If I show you the face first, you don't know anything about him yet. But if that starts to predict an outcome like this, then clearly he's the CS, or conditioned stimulus. And so clearly in this scenario, this plays the role of, you got it, the United States. Seriously, that joke's funny. Okay. It's the unconditioned stimulus, right? Seriously. And when you run this study, you find amygdala does track this. And different parts of the amygdala track the different people. There's one area that doesn't like the negative guy, and another area that's interested in the positive person, and another, and those areas still are higher than they are to the neutral person. So this is part of what your amygdala does. It's also a social Pavlovian conditioning paradigm that you can only do in humans, or can you, right? So we're going to try it. It could work. All right, by now you're probably asking yourself, can or why would anyone base an entire career on one little tiny almond area in the brain? Certainly the rest of the brain does something. Certainly you couldn't be a brain scientist if you were going to ignore everything else. And I'm here to tell you, you can. You can just ignore it. You can just say, I have two questions for the brain. What does amygdala do for you, or what do you do for the amygdala? And that's pretty much all I've done with my career. I recently have begun to consider at least one other area of the brain. So we'll mention the prefrontal cortex in this context. We'll mention it because they're connected, and we know they're connected. We know the axons from the amygdala go to prefrontal. We know the axons from prefrontal go to the amygdala. So we know they can be talking to each other. But the question is, if the amygdala is so good at what it does, and it can automatically pick up on these things and get you going, why does it need a prefrontal cortex? It seems to be doing pretty well on its own. Well, the answer to the question is that signals in the environment aren't always that clear. Sometimes areas of the brain like the amygdala are going to need help. What if you get a face that looks like this? While everyone who runs in our studies agrees that the fearful faces are definitely negatively valenced, we get differing opinions about this expression. Some people tell us about a car coming at her, and other people tell us about her walking in on a birthday party. That is, some people decide to interpret this